Hello, I would like to welcome you on our next series of presentation regarding the application of stem cells. And we stopped last time while discussing the accessibility of the systemic application of mesenchymal stem cells in humans. And we have again Dr. Roger Schechter. He is a key opinion leader in wound healing. And here we are, Dr. Schechter.
Well, thank you, Dr. Rahmo. At this point, I'd like to ask you a few questions because I'm interested in small mobile stem cells and how they can be applied to various medical conditions and also for regeneration.
Yeah, I think we understand that there are some deficiencies observed with the application of mesenchymal stem cells, and these are serious deficiencies. We spoke in last time about the limited viability of mesenchymal stem cells inside in vivo, especially if we are talking about allogenic mesenchymal stem cells that come from different individuals. So these type of cells tend to actually accumulate in the lung, and they rapidly die. It's probably 24 hours or 48 hours. Most of the cells will be dying. There are also other deficiencies related to the size. These cells could clump, and they could actually be hindered within the blood circulation. We have the blood capillaries that are extremely narrow. So these cells are actually not really made to inhabit the blood circulation. They rarely actually move to this tissue. So we are quite happy to say that contrary to that, small mobile stem cells have a small size, much smaller size. They have the ability to travel within the blood circulation, and that's where we actually isolate the cells. So it's a natural habitat to these cells, and we expect them to survive way more easily within that habitat.
Just out of curiosity, you were talking about the clumping of the mesenchymal stem cells. Is there a difference between mesenchymal stem cells, cell surface protein expression versus small mobile stem cells?
Yes, definitely there is a big difference. One of the things that we have observed clearly is the fact that these cells have quite less or actually absence. In most cases, there were no major histocompatibility proteins. So we don't expect agglutination or attachment to immune cells. And in addition to that, we did not find any proteins related to cell-cell adherence, the typical ones that are quite common in creating strong cell attachment, intercellular attachments. We know that SMS cells can attach strongly to certain surfaces and do attach to certain cells, but we do not observe these intrinsic tendency to attach to each other under most conditions. The one that we observed quite often or almost always with mesenchymal stem cells.
I see. So in addition to their difference in size and difference in cell surface proteins and propensity towards agglutination. How are SMS different in their overall constitution or structure and contents versus mesenchymal stem cells?
It's clear that SMS cells have distinctive properties in addition to dimensions, and these are mainly related to their physical and chemical stability. We observed quite extraordinary resilience towards physical hardships, and in addition to that, chemical resilience, like for example pH changes, had little effect on the cells. This is quite important because handling a cell in an industrial setup is often quite challenging if you have a sensitive cell that can be compromised in its quality.
So what is it about the structure of a small mobile stem cell that renders it more robust than a larger cell such as a mesenchymal stem cell?
Yeah, that's an excellent question. I think we're still exploring that, so we don't have a definitive answer on that yet. However, I can say that all our observations are consistent with the fact that we have a very sturdy shell, an envelope that surrounds the cell and probably causes this sturdiness, in part at least. We suspect other chemical constitutions that are specific to the cells that provide the cell additional chemical stability. For example, we observed a large amount of heat shock proteins, and we know that these type of proteins have a big advantage in maintaining the structure and the conformation of proteins under different and various conditions.
Are those intracellular proteins or are they extracellular?
They are intracellular proteins.
So when you were mentioning a extracellular rigidity or cell, external shell,
yes.
What is that made of? Is it some type of extracellular polymer in a sugar form, or is it protein structure?
I'm not going to comment on that right now, but you're very welcome to ask. Yes, I think there is a potential for a sugar coating that can contribute to the sturdiness of the cells. But I'm not going to confirm that right now. As I said, this is still part of our research. We know that that these questions are quite important, but we do also have to set priorities, and the priorities that we've been setting in research is directed towards the experimentation that relates to its therapeutic value.
I see. I realize that that's a tangent of discussion, but it is fascinating because most human cells don't have an extracellular polymer, saccharide-based, and that's more of a characteristic of bacteria or plant cells, and so it really is interesting and speaks to the uniqueness of these cells.
Yeah, as I said, I did not confirm the existence of this sugar coating. It's true that we do have have features that are not typical to the large cells that we observe in the animal, or we used to observe in the animal and human organism. But we know also that humans and animals as organisms are quite resilient as a whole. And it would be quite surprising if that resilience was not reduced to some type of cells that are the source of that resilience. So it's for me hard to imagine that in a resilient organism overall you will be building such a feature with very sensitive and weak cells, especially stem cells, because stem cells should be these cells that come into action when the body is in most need of new cells and repair.
If you're utilizing small mobile stem cells for treatment, why might they be more successful than other stem cell options or other cell therapies?
Yeah, this is a very important question. I guess in addition to the features that we mentioned, one important aspect of these cells is their ability to proliferate extracorporeal in vitro within a dish in almost indefinite manner. So this is very important because currently adult stem cells have the deficiency of changing their property during in vitro amplification or cultivation. So we need to create a donor-independent source for the cells that are used therapeutically. We need these cells to be able to generate a lot of cells without sacrificing the quality and the performance of these cells. So that is an important aspect of industrializing regenerative medicine. And these cells provide this opportunity quite more easily than any other adult stem cells we came across. So that's an important feature. Another feature, I think, is related to how we expect the immune system to react to these cells. We need cells, if applied allogenically, would not cause a strong immune reaction, or preferably any immune reaction. And we suspect that these cells have the ability to do that based on the constitution and some animal experiments that we have done and that we continue to do.
Any idea why small mobile stem cells are so robust that they don't change phenotypically with each passage or generation or cloning, whereas mesenchymal stem cells lose their phenotype after one or two passages?
Yeah. Yeah, I think I have to hypothesize here, and we have some ideas about why that is a fact. A cell like SMS cells that we suspect to be involved in tissue repair has to be adapted to harsh conditions. And a cell that would actually visit a necrotic tissue would be not served well if such a change in the environment will compromise its performance. So we expect these cells to be quite tolerant to surrounding conditions. And based on that tolerance, I think in vitro conditions might be actually a big change for any human in vivo cell, but that change is not enough to cause a continuous deviation in quality and performance as we observe with the larger cells. Now, part of that is also the fact that these cells must be simple, must be designed to be less demanding than the larger, way larger mesenchymal stem cells or comparable stem cells.
Interesting, because you're mentioning necrotic tissue. I do wound management, and one of the concepts of development of new capillaries and chemoattractants for endothelial cells in order to bring about angiogenesis is the hypothesis of a hypoxic gradient, that within the center of damaged or necrotic tissue there is poor oxygenation because there are a paucity of capillaries. And as you move to the periphery of the wound, then there's improved oxygenation because there's improved blood flow through the capillaries. An interesting study would be to see if small mobile stem cells are attracted into an area of hypoxic gradient, even an artificially produced wound, whether or not they tended to migrate to the area of lowest oxygenation. One mechanism in the past that was posited for angiogenesis and attraction of endothelial cells was that nitric oxide being released due to the hypoxic gradient was a chemoattractant for endothelial cells. But the question is, where is the nitric oxide coming from? Is it possible that that may be a cell signaling molecule released by small mobile stem cells?
I think we need to reserve a new episode regarding angiogenesis and the involvement of small mobile stem cells. And I think that's a quite interesting topic where we can continue later with intense effort in trying to understand how SMS cells can contribute to angiogenesis within wound healing within the tissue.